Hello everyone, this is Ridhima. I am a postdoc in Dr. Jimmy Holder's lab at Baylor College of Medicine, and the title of my presentation today is Characterization of Human iPSC-Derived Neurons with SYNGAP1 Mutations. At the excitatory synapses, SYNGAP1 has a potent role in regulating synaptic development, acting as a major player in behavioral and cognitive maturation. It is one of the major genetic risk factor for global developmental delay, autism spectrum disorder, as well as epileptic encephalopathy. Pathogenic loss of function variants in SYNGAP gene are causally linked to neurodevelopmental disorders defined by cognitive impairment, social communication disorders, as well as early onset seizures. SYNGAP pathogenic variants are surprisingly common, with the incidence reported in as 1 to 4 per 10,000 individuals, which accounts for about 1% of the sporadic neurodevelopmental disorders. Previous studies in mice have reported that SYNGAP1 heterozygosity results in hyperexcitability in the brain network, resulting directly affecting cognitive functions. Although rodent models of SYNGAP haploinsufficiency help uncover the pathophysiology of underlying neurodevelopmental disorders, these models cannot fully recapitulate all aspects of human brain development and dysfunction owing to genetic complexity, protracted time of development, and intricate cortical expansion of the human brain. Thus, with an aim to better understand the pathophysiology and functional consequences of SYNGAP1 inefficiency, we want to characterize SYNGAP mutations using human iPSC-based model system. The human-induced pluripotent stem cells, they serve as a comprehensive model system to study early developmental stages, and when derived from the patient-specific sources, these cells, they serve as a powerful tool for disease modeling. And patient-derived human pluripotent stem cell lines, they have a unique capacity to model human disease in a dish. In order to study SYNGAP1 pathophysiology in a human-based model, we collaborated with the Rumbog lab at Scripps Institute, Scripps Research, Florida, and they used CRISPR technology and successfully created isogenic human-induced pluripotent stem cell models for studying SYNGAP pathogenicity, and we aim at subsequently using this as a tool to discover SYNGAP mediated phenotypes in the patient-derived neurons. So they targeted exon 7 here, and they chose exon 7 because it's one of the common exon presence in most, if not all, SYNGAP transcripts. And also in the rodent models, SYNGAP deletion has shown to cause SYNGAP protein disruption. So they created two two homozygous edited SYNGAP knockout clones along with their unedited isogenic wild type using a standard human iPSC line to produce these clones. And they also looked at the protein expression. So SYNGAP protein expression, it was almost disrupted in the knockout clones versus the isogenic SYNGAP1 wild type clones. So once they have that model system in hand, they performed some morphological characterization because from the rodent studies it has been shown that SYNGAP1 loss of function, it disrupts the maturation rate of dendrites and synapses. Therefore they examined the dendritic morphogenesis in developing induced neurons produced from each of the 4 human iPSC clones. And they looked at if we look here, the knockout ones on the right, they clearly show they display larger dendritic fields compared to the wild-type clones. And after the measurement of the length of the primary, secondary, and tertiary dendrites, they see that the knockout clones, they display larger dendritic fields compared to the wild-type clones. However, in contrast to the length, the complexity of the dendritic arbors, it was not affected by SYNGAP disruption. And then they also performed some immunolabeling of the dendritic as well as synaptic proteins. And clearly from this here, so the right panels are for the SYNGAP knockout compared to the wild type controls. So the immunolabeling studies, they showed enhanced expression of MAP2 in the knockout neurons. And it was not just also shown here, the quantification here, but it was not just because of the higher density of the knockout neurons present in the cultures. Even when this measure, even when they did this measure was normalized to the soma count, the effect, it was larger for the knockout neurons compared to their wild type counterparts. So SYNGAP disruption results in larger dendritic length with more postsynaptic structures. They also looked at the PSD95, GLUA1, where it's seen here clearly that the knockout neurons, they display much more increase or like enhanced structures at the postsynaptic region. Next, in order to look how the reduction in SYNGAP expression affected neuronal network activity, we employed MES system, the microelectrode array system, to record spontaneous network electrical activity from the knockout as well as the control lines. So here is just a bright field image of 1 week old neurons on an MEA well. And these are the representative waveforms of the spiking behavior from each of the wild type as well as the knockout neuronal cultures. And here we can see from the representative raster plots, these are the representative raster plots from all the 4 clones, which are illustrating the spiking and bursting behavior over a 5 minute of continuous spontaneous recording. And if we compare the raster plots for the knockout cultures to the controls, they clearly show that the spiking was significantly higher in the knockout cultures, with even the network bursting shown here in pink. And yeah, so here these are the we quantified the mean firing rates for all the 4 clones here individually, as well as then grouped together by genotype, along with a longer developmental timeline starting from week 2 to week 6 of the neuronal maturation. And the mean firing rates are clearly like higher for the knockout versus the wild type control lines. Yeah, so the spontaneous MEA recordings from the knockout neurons, they exhibited significantly increased activity when we compare them to the wild type controls. Next, we also looked at the excitability of the neurons, which is observed as the ability to fire multiple action potentials within a short time period. And we quantified the average number of bursts, and here are just the plots showing the cumulative plots showing the average number of bursts for the individual clones up here, and then grouped together by genotype. And the bursting patterns for the knockout neurons were much more robust compared to the wild type isogenic controls, indicative of hyperexcitability in the neurons having disrupted or null SYNGAP1. Next, I also quantified the network burst to quantify the synaptic connectivity. And these are again the cumulative plots of the average number of bursts for all the clones individually and then grouped together by genotype. So the knockout neuronal cultures, they display, as it is seen here, the knockout neuronal cultures displayed different degrees of neural network activity observed as a network burst, which is accounted here, as early as like at week 3 of neuronal maturation. Thus, we can say that SYNGAP expression, it has influenced, or it influences the dynamics of cellular activity in the developing neuronal networks. Next, we aim at extending same methodology from this like engineered neurons or the engineered cell lines or the model, and we want to extend the same methodology to characterize the effect of SYNGAP1 loss of function in the patient-derived neurons. So we employed one of the patient lines. So here is just the workflow of generating the IPSCs from the patient blood, along with the karyotyping and other quality control here. And then we engineered, we used CRISPR-Cas9 gene editing of the SYNGAP patient line, which we call as S3059 line, and we created in order to create the isogenic control. So this is the patient line with the single point mutation in exon 4. Here it's confirmed by the Sanger sequencing, as we can see here. So there is this A has been deleted among the stretch of this C's and G's. However, using CRISPR-Cas9 we use the sgRNA with the sequence shown here, along with a single-stranded DNA repair template. So we use this technology to correct for this deletion, and this is the Sanger sequencing confirming the corrected sequence with the incorporation of A back in the region of interest. So next, we also confirmed the edited iPSC clones after we corrected them. We also confirmed the correction using the next generation sequencing. So as we can see here, the number of reads and just like look at the percentage of the number of reads for the S3059 heterozygous mutations with a deletion of A in this region of interest. This is from S3059, the parent line, which we used or employed for correcting to get the isogenic clones. And here are the two, we have been successful to get like two isogenic clones. And if we look at the percentage reads, they have already dropped down to 0, and we don't have any deletion in the region of interest. It's almost like 0. And we are also trying to confirm any effects of any off-target effects, and we have sent out the sequence, the samples, the DNA samples for whole exome sequencing, but we are still waiting for the results and hoping they are good, just like we confirmed with the NGS. So in summary, this study provides an in vitro model system to study mutational effects of SYNGAP1 and SYNGAP deficiency or haploinsufficient, SYNGAP deficiency, it causes increased neuronal excitability as depicted in the morphological studies along with the functional MEA data. So the main results showed that the knockout neural cultures displayed coordinated neural activity at a network level, indicating their early on maturation compared to the wild type controls, which further supports the idea that SYNGAP1 dysfunction causes cognitive abnormalities in the patients with SYNGAP1 inefficiency by possibly disrupting the neuronal networking. So we have employed and we aim to extend our methodology using the human iPSC-based neuronal model to employ this model of SYNGAP pathogenicity to use it as a tool to discover the SYNGAP-mediated phenotypes in the patient-derived neurons. So we have already started working with S3059 line, which we have already been able to correct to create the isogenic control. And then we have the BCMJH17 line, which is a splice site mutation. And we want to characterize how this model can be used to study the SYNGAP patient phenotypes. Lastly, I would like to thank Rambog Lab, who engineered the SYNGAP1 null knockout clones and performed the morphological analysis. I would like to thank uh, Jean Kim, as well as my PI, Doris Duke, Charitable Foundation, as well as SYNGAP Research Fund. Thank you.